possible.
But maybe someone else. Maybe someone else. But maybe actually something that is possible responsible. Anyway, I'm just a rubber job. I still live in 5 years.
So if you put the three together, and it's no. So you can escape the band in W2. You just can't do it. So that was the part that I was. You're too smart. You're too smart for that. That makes sense.
Animal hand lotions.
I was like once, but I disregarded it for some reason. So I extended my friends by
DNA might go.
And also, I was doing there because it's like this is very sad.
it was because we were
No, the one that was lost. Yeah, and that was one of the general 149, and everyone else had to be there. Oh, it was, yeah, it was pretty good overall.
Oh, like even how you
It
basically
was, but it was mostly good because of all the complementary. It's not so much as the actual. Oh yeah. It's like nobody Spanish. Yeah. It's like it's kind of a big robot.
My main.
And then the top.
Do my email.
Those goddamn things are around here all the time.
What time do you have to pick them up?
Yeah, I guess you can choose. I think we're doing improvements right now before it's going to start in May.
I also applied to
Make sure the light. Just look at that. He's supposed to be responding actually not on hard copy.
Yeah.
So Wario seems to come out with a good product.
I think my staff
Yeah. Well, I think it's just a nice user interface.
I haven't seen it.
Yeah. I mean, it's your average person that wouldn't care that they don't care if it picks the right articles as much. They just care if it's really flat.
So it's a nice interface.
Yeah. It basically just shows you like little newspaper articles, and they're all like custom selected for you, and you can scan through them, and then the layout is nice. And then if you click on an article, it pops up the article right here.
2 years ago.
And then you can select sections. So you do it like this one, like that one.
Oh, I took 320 in summer after
Like
first year.
sections, so do like one article. Technology.
Oh, I did 121 in term 2, and then
Yeah.
221 in the
They have a lot
first
of
term of summer, and then I was like,
I haven't actually
I don't understand the story. Well, we got scaled a lot, because the questions were very hard, and
So hopefully,
the average
I
was
think it's
50 or even lower, and then we got on the midterm.
they have there's already one other competitor who does sort of similar thing.
Oh, it was a basic lecture from and his area was photography, so we had some interest in photography.
Just the Google News web, it's not really flashy, you know.
So it is with the greatest pleasure imaginable that I'm introducing to you today Anne Condon as our very special faculty lecture speaker. It's with particular pleasure that I do that because Anne has not only been a great colleague for many years, but also an extremely valuable mentor to myself. So I was very pleased when I was asked to do this today. I'm also a little bit intimidated by the fact that Anne herself is the person who probably does the most graceful introductions you could possibly imagine, and I feel like I can't possibly rise to that standard. However, here I am trying to introduce her. So let me tell you a little bit about Anne's background, not too much, then about her research, and then we'll go, of course, into what you really came here for, namely Anne talking about her very exciting work. So Anne did her Bachelor of Science at the University College Cork in Ireland in 1982. And I think Anne was also born and raised in Ireland, so that's kind of where her roots are. Her PhD then was from the University of Washington in 1989, and I guess this is one of the points where it became already quite obvious what a special person Anne was, because that dissertation was rewarded with the ACM Distinguished Dissertation Award. She then moved to the University of Wisconsin to become a faculty member there, where she stayed for the next 12 years until 1999, when we managed to get her here. I can take zero credit for it. I happened to be around and benefit from her arriving. I remember the day, actually, I believe, when she first came here for anything other than just getting set up. And ever since then, she's been doing tremendous work here at UBC. So in particular, she had the NSERC General Motors Chair for Women in Computing between 2004 and 2009. She was then an Associate Dean for the Faculty of Science. Actually, that overlapped even between 2007 and just last year. And she stepped only down from that job because she agreed to become the head of computer science and will start in that role in July this year. So currently, my understanding is that Anne is technically on leave, which makes it particularly nice of her to come to speak to us today. Anne's work has been recognized with many awards throughout her career. I already mentioned one, but there are many others. She got the NSF Young Investigator Award in 1990 and the University College of Cork Distinguished Alumna Award in 2001. In 2006, she was a Distinguished Scholar in Residence at the Peter Wall Institute here at UBC. And still is a member of the advisory committee, which is sort of a small and select group that steers that institute. In 2010, Anne was recognized with the Computing Research Association's Nico Haberman Award for outstanding contributions that had to do with her engagement in promoting the success of women in computing and other minorities. And this is a very important aspect of Anne's work that sort of runs through like a thread. It also played a role in her 2010 ACM fellowship, which she got awarded then, which also recognized her contributions in complexity. And so, in terms of research, there is a lot to talk about. I'm not sure what exactly Anne will be touching on today beyond her work in computational biology, which I think is where a lot of her recent energy has been. But Anne has done an astounding amount of work in complexity theory, starting from the investigations into the power of randomness and non-determinism, then including things like stochastic games, probabilistic planning approximation algorithms, and proof complexity, just to mention a few. She has also worked on verification, in particular cache coherence protocols, and has been doing some very well-recognized work in computational biology, particularly in the area of DNA and RNA design and DNA computation, which is very close to what she will be talking about today. This work has led to a number of very, very prestigious publications, including the prime venues for the life sciences, namely Science and Nature, and has made Anne to one of the world's leading experts in this particular and very exciting area. What this area exactly is about and why it is so exciting, Anne will tell us in her talk. Thank you very much, Anne, for being here today and for talking to us.
Okay, wow, it's going to be hard to live up to that introduction. Today I'm going to talk about how you can program molecules and why that's interesting. It's a field that I've been following for about 15 years when it got started through work of Len Adelman. And it's a field that's very intriguing to me. As a first illustration, I chose this little smiley face, which was designed by Paul Rothman in 2006. It's better. So the smiley face has a width that's about 1,000th times as narrow as the width of a strand of human hair. It's made completely out of DNA. And it's an example of molecular programming because the principles that Paul Rothman used to design it, namely scaffolded DNA origami, could be used to generate any pattern you want. In fact, you can find many other nice examples on Paul's webpage. This is an atomic microscope image of one of his designs. I like the smiley face as an illustration because I think it brings across a key point about the field of DNA computation. And that is that molecular programmers see DNA as a material that can be used in ways that have nothing to do with its use in nature. And this really distinguishes the field of DNA computing, say, from synthetic biology, where people will insert new genes into genomes of living organisms to do new things. It's also very different from the field of bioinformatics, where the goal is to understand the molecular mechanisms of the cell. Here we're starting with a material DNA that happens to come from the cell, but we may do totally unrelated, interesting, and creative things with the molecule. So since it's a very creative field, I thought I would get your creative juices flowing by showing you a very short clip of a video called The Inner Life of the Cell. And this video shows molecules in action in the cell. So while you're watching it, I want you to think about what those molecules are doing and what they suggest about the potential for molecules to perform interesting computations. So keep your ideas, keep track of your ideas, because as soon as the video is over, I would like to hear some of your ideas about molecules and their potential. So I'm going to turn out the lights just for the video. Isn't that cool? It reminds me of the idea that while energy permits things to exist and to act, computation permits things to be purposeful. Okay, so what things in that movie remind you of computation? Suggest computation. David?
Like a Turing machine.
Great, looked like a Turing machine. A man after my own heart. Great. Other specific things? Holger.
Well, we saw replication, right? And we saw
replication.
programmed recognition of things.
Programmed recognition of things, good. So pattern matching, recognition of things. The motion of that little walking thing, right? Right. Yeah, like a little robot. Sort of have these visions of a department head carrying the weight of the world on their shoulders, and I see that. Excellent. Anything else? All right, those are great examples. Okay, so sort of following some of those themes. Let's go. I'm going to structure my talk in 3 parts: sequence, structure, and pathways. So these are 3 ways that suggest how we can program DNA molecules. Sequence is kind of pretty obvious. A DNA sequence is made up of 4 letters, A, C, G, and T, which we can use to encode information. Structure, like this beautiful structure here that we saw in the movie, is a way to organize information. And with DNA, we can use structure to organize information at the nanoscale. There's many other interesting examples of structure in the video. In this example, that organization helps, for example, to channel the way other molecules are transported around the cell. Structure forms in a DNA molecule when it folds on itself and bonds form between pairs of bases. So we'll see that you can program structure and you can do interesting things with it. And then finally, pathways. Folding pathways of a molecule are sequences of structural changes over time. And as a structure changes, that enables molecular movement. So in this example, bonds are changing between this little walker and its track, enabling movement. Similarly with DNA, we can arrange for molecular movement. And molecular movement then permits us to control how things would move at the nanoscale. Also, it will help us be able to design systems that can respond and change as a result of changes in their environment, for example, which is very useful. So for each of these, how to program molecules, I'll give examples of why I think it's interesting to program molecules these ways. And I'll tie it in a little bit with the research that I've done over the past several years. This order of presentation also roughly follows the order in which the field has evolved, the way ideas in the field have evolved from sequence to structure to folding pathways. Okay, so let's start with sequence. This little picture here shows a sequence which is being translated to produce some kind of interesting protein or something like that. Okay, so the first person to actually go into a wet lab and write a molecular program was a computer scientist called Len Adelman. Why was Len interested in programming molecules? Well, he was sort of awed by the remarkable energy efficiency that at which things happened in the cell. So he saw the potential for energy efficient computation. He was also impressed with the information density that you can achieve if you store information in DNA molecules. 1 bit per cubic nanometer, that's just like way orders of magnitude smaller than anything we can achieve with conventional technology. And finally, since we can have many strands in a test tube, we can do massively parallel computations on these strands. So these were the ideas that inspired Len Adelman. But when I read about this work, I also thought it was very cool. And I decided that if I was going to work in this area, I was a theoretician, still am a theoretician, but I wanted to actually, work with people who had a wet lab and do some cool stuff and not just prove theorems about DNA computing. So I had the good fortune of collaborating with Lloyd Smith and Rob Korn at the University of Wisconsin, where I was at the time, and their postdocs and students. Rob is a chemist. He's a surface chemist, so he specializes in figuring out how you attach things to planar surfaces, like a centimeter square silicon chip, for example, or glass plate. And Lloyd is interested in DNA and particularly interested in working with DNA that's attached to surfaces in the way that Rob can do. So they said, okay, we're happy to think about how we can compute with DNA, but we want to do it on surfaces. Okay, so what does it mean to compute with DNA on a surface? Well, you take your little planar glass plate or whatever you have, and you use Rob's technology to tether DNA strands onto that glass plate. You can think of them like strands of grass growing off the plate. You could put 10 to the 12 such strands on the centimeter square chip. Once you've got them on the chip, of course, you can design them before you put them on the chip, and you can design them to encode information. This is a big simplification over what we actually did, but it gives you an idea. So in this example, each strand is encoding 3 bits of information, and each bit is encoded by 3 bases from the set A, C, G, and T. Okay? And I just happen to have 8 distinct strands, which would be all the 3-bit sequences. Does that make sense? All right. Well, you've got now this information on a surface. I should have said that the reason they like to work on surfaces is that it gives them a lot more control over what you're doing. Because if you do something interesting with your DNA on the chip, then you can wash everything off, like if there were enzymes that you needed or something, and start again with a completely clean environment. Whereas if you had things in a test tube and you put some enzymes in, it's very hard to get all the enzyme out to do another thing later on. So that's the reason we're working with surfaces. So we thought about how we might compute with information encoded in DNA strands and surfaces, and we came up with 3 operations that Lloyd thought they could implement in the lab and that we thought would be useful to do something. And those were these 3 here. So the first operation lets you mark strands based on their information content, so select strands based on the information content. So specifically, you might want to mark all strands that have a particular bit set to 1, for example. And the way we mark strands is simply to make them double-stranded. Okay? So a DNA strand always likes to bind to its Watson-Crick complement to form a double helix. So there are ways to mark strands in this way. Then you can selectively destroy the unmarked strands. You can do that by putting an enzyme near the chip that will chew up the single-stranded molecules. And finally, you can unmark your marked strands, and you can do that simply by raising the temperature, which causes the double-stranded helix to break, and you can wash away those blue double strands. Okay? And then, of course, you can repeat this process. So those were 3 operations that Lloyd and his students, including Qinghua Liu, were able to get to work in the lab. So then we thought, okay, we can use these operations to solve a computational problem. We had a slightly more complicated example than the one here, but it wasn't a whole lot more complicated. So for today, we're going to look at how would you decide what truth assignments satisfy the satisfiability instance up there, the Boolean formula. Okay, so what we'll do is we'll process each of the clauses in turn. We'll destroy any strands that fail to satisfy that clause. So by the end, after we've processed all the clauses, only those strands that satisfy the whole formula will be left on our surface. Okay? To process the first strand, we'll simply to process the first clause, we'll mark strands that have bit 1 or 2 set to 1. Those are the good ones. Then they're marked on the middle on the right there. Then we'll destroy the strands that are unmarked, the strands in which all bits are set to 0, or the strand in which bit 3 is set to 1 and the other 2 bits are set to 0. We're left with 6 strands. And then we'll unmark those so that we can move on to do exactly the same thing for the next clause. We repeat that process for clause 2. We'll mark all the strands that have bit 3 set to 0 because those are the good clauses that are continuing to satisfy the formula. We destroy any clause that's not marked. Those are the bad clauses. And we unmark at the end in case we need to go on and do some more processing. At the end of this process, what we've got are exactly those strands that correspond to truth assignments to those bits 1, 2, and 3 that satisfy the formula. And then Qinghua Liu developed amazing technology for reading back out what exactly those were, which was actually one of the trickiest parts of the whole process. Okay. So that's the way we did a very simple DNA computation. Okay, so now you see how we can program DNA molecules using their sequence. Why would anybody want to do this? Well, at the start I explained why Len Edelman was optimistic or had some hope that we might be able to do energy efficient, massively parallel, high information density computations. Well, in actual fact, these tiny demonstrations didn't offer a lot of promise, and indeed, 15 years later, I would say nobody is overly optimistic that we're going to solve really complicated computational problems using ideas like I just presented. So I just put up 2 quotes from Len and from Lloyd that sort of suggest, well, this is kind of cool, but maybe it won't scale up to do anything really astounding. However, I do think the ideas have some merit from a practical standpoint. People like Sidney Brenner and others have suggested and in fact have labeled molecules in a chemical library with DNA tags, because DNA is such a nice molecule to work with, and you can do all these kinds of cool operations on DNA molecules. So often, say for example, a drug company or a drug researcher might have a huge library of proteins or some other chemicals, and they want to find which one is going to bind to a pathogen and possibly be a good therapy for a disease. Well, it's very difficult if you have such a library to figure out which molecules have certain properties and which ones are doing certain things. But if you tag them with DNA, you can use the DNA tag in some sense to do the information processing part, while the protein part will be working, binding to the pathogen. Again, it's not like anybody's really harnessed DNA computing to do this, but I do think it's a good rationale for why ultimately it might be useful if we get more control over our DNA operations. Okay, so to summarize from the first part of my talk, Edelman had this great idea that maybe we could compute with DNA, and I presented just one example of a project I was involved in, but there were many other exciting projects that people worked on in those early days of DNA computing. It's true that computing with these kinds of sequences hasn't shown promise for solving purely computational problems, but there are parallels with the way people use DNA in a field called encoded combinatorial chemistry. And finally, this early work paved the way for programming with structure and folding pathways, which is what I'm going to get to next. So that was another kind of outcome of these early period. Any questions before I move on to structure?
So the readout part, I guess you didn't say much about it, and I understand why, but is it reliable at all? I mean, can you detect? Are you getting probabilities out, or do you have to replicate the same kind of tests to get some kind of statistical sense of?
Oh, it was actually quite, conceptually quite simple and easy once it worked in a limited context. Okay. So the idea is that once you're done with the computation, you can actually elute those strands from the surface, and you can expose them to something like a DNA microchip. Okay. And the tags are fluorescently tagged, and then you read the fluorescence intensity off of the chip. So then you can see which spots light up on the chip, and that tells you exactly which spots are big. And in our experiment, like you look at the image, it's really obvious. You could just tell by eye that wasn't, but this was a very small experiment. Yeah,
this trial operation, did somebody actually put something in there in the chip, or was that something that could be automatically done?
Which part?
For example, in the destroy operation?
Well, I think in this experiment it wasn't completely automated, but it wouldn't be hard to completely automate it using, for example, microfluidics. Oh. But in this case, it wasn't. Oh. So there was a manual step. Yes?
And what about data input?
Data input, well, so you have to synthesize the molecules to begin with. In this case, we had to synthesize a combinatorial set. And standard DNA synthesis techniques are amenable to that kind of thing. So we didn't have to, if you had to synthesize each strand independently, that would be a disaster, right? But you can synthesize them in a massively parallel fashion. And then we attached them to the chip that was, just sort of en masse. So there was no need to put a specific strand at a specific location. You could just go anywhere. So that part is again, in principle, scalable. Okay, so we'll move on to the second part of the talk, which is structure. So let's start with what is DNA secondary structure? Well, it's the set of bonds, either AT bonds or CG bonds, between bases of a sequence or possibly multiple sequences. You're probably all familiar with the DNA double helix, which is when we usually see these complementary bonds between one strand and its complement, right? Well, if you don't have two complementary strands to form a double helix, say you just have one strand, bases in that strand are happy to bond with other bases within that strand, creating structure. Okay? So on the left, I have a very simple structure. There are 4 base pairs between 4 C's and 4 G's. If I were to draw this more realistically, it would be like a tiny little half double helix or something, but I don't want to try to draw those twists, okay? And you've got a little loop at the end there with a bunch of A's in there, and something like a simple structure like that is often called a hairpin structure. I'll probably use that term a lot as we move forward. The structure on the right would be an alternative for that same sequence. The sequence is the same, but the structure is different. In this case, all the A's are binding with all the T's. Okay, so sequences could assume many different possible structures. I've given 2 examples here. Everybody see what a structure is? All right. Here's a more complicated structure designed by a chemist called Ned Seaman at NYU and his students. He's been working on designing DNA nanostructures for quite a few decades now. This particular structure is called a double crossover structure. You can sort of see that there's 2 crossover points in a natural way here. It's made out of many different strands rather than one strand in my first example. So I've colored the strands so that you can see where different strands start and end. And the little black straight lines that are barely visible are the base pair bonds. So I didn't put in all the ACGs and U and T's, but you can each one of those little lines that connects one strand with another sort of corresponds to a base pair. And then there are 4 ends where there are bases that aren't bonding to anything. Those little black lines are just sort of hanging there, not bonded to anything. Okay? And I've labeled them A, B, C, and D. In our field, we call those 4 little things sticking out sticky ends because they are willing and available to bind to something else. I mean, bases like to bind with their complements. So they're ready to bind. Abstractly, we can think of this thing as like a tile. It doesn't really matter what the inside part is because it's not going to bind with anything else. Everything is bound up. But the 4 sticky ends are available for binding with other things. Okay? So we can use that abstraction and think about tiling theory for mathematics, for example, and how you can do computations with tiles and maybe compute with these little structures. That's the idea. Okay, so how would you do that? Imagine that you have a bunch of tiles and they have these little sticky ends. So here's a new tile, and it's got sticky ends that are complementary to some of those in this little assembly that's building up here. Well, if it can bind to places that would result in extra base pairs and, again, lower energy for the overall set of molecules, then it will do so, which is what you see here. Okay. And if it didn't match, then it wouldn't bind, and it would just sort of float around. Okay. Okay, so a student at the time that Edelman did his work, Eric Winfrey, thought, well, maybe instead of programming with sequence like Len Edelman did, I could program with structure. So he knew of Ned Seaman's work, and he spent some time, he was out in California, but he would spend some time in Ned's lab out in New York. And many years later, after he graduated and did a lot more work, he finally was able to program something using this principle of self-assembly tiles. And he chose to do a Sierpinski triangle because he wanted to demonstrate that you could program things that weren't periodic, kind of boring periodic things. He wanted to show you could really program with these tiles. So he designed tiles that would come together in a programmed way because of the way he carefully chose the sequences on the sticky ends. And then he synthesized those tiles, put them in a test tube, and used atomic force microscopy to see what the result of the assembly was. And as you can see here in these pictures, you kind of did get something that looks kind of like a Sierpinski triangle, admittedly, with a few errors sprinkled in. Okay, so that is one way that you could use structure to program DNA. Why would you ever want to do that? You don't really need nanoscale Sierpinski triangles, right? Well, people have various ideas why it might be useful. One idea is that if you can build regular structures like lattices out of DNA at the nanoscale, they have nanoscale resolution, you could use them to position other molecules in a precise way at the nanoscale. So, for example, one group, John Reif's group at Duke University, produced the work on the left-hand side here, where they took protein molecules and were able to tether them to the points in a lattice and arrange them there, therefore in a regular way at the nanoscale. Another researcher in the field, William Shih, has been very interested in designing DNA scaffolds onto which, again, he can position proteins. He's interested in transmembrane proteins, and he wants to be able to take X-ray diffraction images of these proteins. And in order to do that, he needs to arrange them in sort of a crystal-like array, many of these proteins in a very regular way. So he wants to build 3D rather than 2D scaffolds, and he's already well on the path to do so, as the picture on the right might convey. He's also interested in building curvature into DNA nanostructures. Okay? So ultimately, his goal is to be able to take precise images of transmembrane proteins or other types of interesting molecules. So these are two examples of molecular programs that are using DNA scaffolds with the hope of tethering other molecules and therefore better understanding those other molecules. Okay, so that's one possible application. Again, something that hasn't completely panned out, but something that researchers in the field are hoping will be useful. Personally, of course, I think there's lots of neat questions about computation, and that's a good enough reason to kind of pursue this kind of work. The Sierpinski triangle work, for example, raises new questions such as, well, how would you correct the errors that arise in the type of self-assembly computation that Eric Winfrey did with his students? Another question that arises, if you want to build something like a Sierpinski triangle, or you want to build a square that has dimension exactly n by n, how many different tiles do you need in order to do that? The fewer, the better, because it'll just save you a lot of effort in terms of design. Okay. So while mathematicians, I think, developed a beautiful theory of mathematical tiling, now computer scientists can think about resource issues, complexity of patterns relative to the tiles. So there's lots of interesting questions here. And there are many other research opportunities, for example, in the design of 3D rather than planar structures, which people in the field are really just getting started on. So next, I'll just talk very briefly about research questions in this area that I've worked on, in fact, closely with some people in the room today, Holger in particular. So I got interested in these problems because of the direction that the field of DNA computing was taking in molecular structure. And at that point, I had left Wisconsin and left my chemistry collaborators, and I was at UBC. So rather than designing structures and thinking about how you might implement them in a wet lab, what I did instead was to get interested in computational tools that might be useful to people who want to design interesting structures. So if you want to design a structure, what that means is you're going to decide what sequence to synthesize so that it will fold into the structure you want. And if you want to be able to do that, you certainly need tools that will predict what structure a given sequence will fold into. Okay? So those are the two problems that I started to work on after I got here. And this was work done very closely with Holger and, as I said, many other people at UBC. So one simple question is, given a molecule, what structure will it form? What bases will bind with what other bases? That's really the question. Okay? Well, of course, you won't know how to approach that unless I tell you a little bit more about the principles of molecular folding. So here I'm going to do it in an extremely simple way to start with. We can formulate this question as an optimization problem. So what we want to do is find the secondary structure, or the base pairs, that would have as many, the structure that would have as many base pairs as possible. So intuitively, molecules, their folding is driven by thermodynamics. Molecules are like couch potatoes. They want to be in the lowest energy state possible. Okay? So think of each base pair as contributing -1 to the energy of the molecule. So the more base pairs it has, the happier it is. Okay? So that's the problem. Now, just to make it a little bit simpler too, I'm just want you to think about structures that are branched. The structure here is a branch structure. What that means is that there are stems or double helices of base pairs that you can sort of make out, and they just sort of emanate from, some central point in a branched fashion. There's no piece off the bottom left that's looping back up to the top right. It's just sort of tree-like or branched. Okay? So that's one way to formulate the problem. And of course, biologists have been interested in this problem for a long, long time. So they've figured out good solutions for this problem. The reason it's interesting to consider first branch structures is that you can always break the structure into two completely separate parts. And if you knew where to break it, and you could find the optimum structure for the first part and the optimum structure for the second part, you could put those two together to get the overall optimum structure, because the two parts have to be independent because of the branching. Okay? So that's the key to solving this version of the problem. And anybody who's taken 320 and a little bit of dynamic programming, given a couple of hours, should be able to figure out how to solve this problem. It's not too hard. It uses very classical techniques from computer science algorithms. Okay, so why did we work on this problem? Well, in real life, the problem's a lot more complicated. So we developed algorithms for more realistic versions of the problem. First of all, we worked with realistic energy models instead of my simple couch potato model of -1 per base pair. It gets much more complicated. Some of the models have thousands of parameters. But the principle is the same. Okay? Well, we had to deal with that. We were also interested in situations where structures aren't branches. I don't want to go into that, but it gets a lot hairier when structures aren't necessarily branched. And another complication is that structures may be formed from multiple strands instead of a single strand, which really does complicate things more than you might believe. So those were some of the directions in which our work on structure prediction went. The first bullet up there focuses on work we did to improve algorithms. So we took the energy model or the thermodynamics that biologists said were the right ones, and we focused on getting the complexity of algorithms better, getting things to work faster and more efficiently. One of the things that eventually you sort of realize when you do this is it doesn't matter how fast your algorithm is. If the energy model the biologists gave you in the first place was not that great, you're never going to get great predictions. So after a while, we decided that actually we could make more of an impact by improving the energy models instead of just making the algorithms faster. So again, working closely with Holger and also Kevin Murphy and a collaborator at the University of Rochester in biophysics, in particular, our student, Mirella Andronescu, who was co-supervised by me and Holger, we worked hard to improve the free energy model and actually got fairly significant improvements there. So that was our work on prediction. Finally, we did work on the opposite problem. So prediction says, given a sequence, how is it going to fold? Design says, find a sequence that will fold into a particular structure. So we were able to use some of our prediction tools to come up with very interesting algorithms for secondary structure design. That work kind of grew out of a class project in our graduate bioinformatics class, and some of the students involved in that project, I guess, were not my students at all, but really great students that we had in that class. I'm not going to tell you any more about this work, but a lot of it and a lot of the tools that we developed are on a website called rnasoft.ca. So if you're curious, you can go on there, type in your favorite sequence and see how it folds, or design. Pick your favorite structure and design a sequence that will fold into that structure. Okay, so to summarize then from the second part of the talk, DNA structures can be programmed. You can build complex shapes and patterns from them. One reason that you might want to do that is to build scaffolds for organizing matter at the nanoscale. Programming structures raises new questions in computation, like how do you correct errors in DNA self-assembly, for example. And DNA programming, because of the need to be able to predict and design structures, I think really did advance computational methods for nucleic acid secondary structure prediction and design. And that's where much of my focus, Holger's focus, and the focus of other people here was. So a little bit removed from real DNA computing, and actually a lot more broadly applicable to biologists as well. Any questions about structure?
So the structure depends only on the DNA sequence or the other agents as well when you talk about?
So our view here is that it only depends on the sequence, and that's, I mean, in the context that motivated me, DNA computing, that's a totally reasonable assumption. It's out of the cell. The work that Irmtraud discussed last week is totally different because there's many other complicating factors, and the DNA or RNA is getting transcribed and folding at the same time. Here, none of those issues arise. So really, the right approach is to think about thermodynamics and model the thermodynamics of folding. Great. Yes?
How does this relate to protein folding? Is it the same?
It's quite different from protein folding. Proteins, predicting protein structure is a lot more complicated than RNA structure. So it's kind of a complicated question. We focused on secondary structure prediction, so we actually, in the end, don't know what the thing looks like in 3D. We just know which bases pair with which bases, which can be helpful ultimately in knowing something about the 3D shape or the function of the molecule. Predicting 3D RNA structure is actually harder than predicting protein structure, and there's very little work in that area. There's a lot more attention to 3D protein structure prediction. And you know, the principles at the very high level are the same. It's thermodynamics-based process, but the details are quite different because of the difference between the molecules. Okay. Yes?
Where do you branch away at all from when there are inadequate quantum chemical models on the chemistry side, which they're inadequate to make predictions of what you're observing? If you just depart from that, see yourselves collecting rules of thumb, inputs and outputs, the explanation is a separate task. It would be nice to have the explanation, but in the meantime, we can set up a set of observed occurrences and then just proceed from that.
Yeah, so that's a great idea, and one that would be used actually quite a bit now for 3D RNA structure prediction, where the first principles approaches are not well developed. Absolutely. Yeah. So for secondary structure, though, really these methods are quite good. So it's good to work. You can always combine them with more your suggestion of kind of having databases of structures and looking at those too. Absolutely. Yeah, actually, Francois Majeur at the University of Montreal is one of the leading experts in 3D RNA structure prediction, and he uses those kinds of methods a little bit. All right, so we'll move on to the third part of the talk, to folding pathways. Okay, so what is a folding pathway? Well, you can see one right there. If I can play that again. No? There we go. Okay, so like I said at the start, it's a sequence of structural changes over time. So what you're seeing here is a molecule that starts out in its unfolded state. It's just Everything is unpaired. It just happens to be organized as a circle. And then base pairs start to form and break and form and break as the molecule is sort of looking around for its couch potato configuration, okay? Minimum energy configuration. So a folding pathway is a sequence of structures that a strand assumes as it changes from one structure to another. In this example, it was from completely unfolded to its stable configuration. When molecules fold, it's really a stochastic process. Things are happening kind of at random, although they're biased towards increasing the number of base pairs or lowering the energy of the molecule. Also, when molecules are folding, I guess they don't like to climb over high barriers in their folding process. So if they can avoid that, they will. They like to take the easy road to get to their lowest energy configuration. Okay? Does that make a little bit of sense? All right. So the particular,
So do they get stuck in local minima, or you're saying they always end up in their?
Oh, sure, they can get stuck in local minima.
Okay.
Absolutely. So the particular little simulation that I showed is off of a web server of Isambert, and it's called the Kinefold web server. Again, go on there and type in your favorite molecule and see it fold. Okay, so I showed you a little movie. I showed it to you twice. It folded the same way both times, but obviously if you really ran it independently more than once, you'd get different folding patterns each time because it's stochastic. Okay, so what I want to latch on to here a little bit, though, is the minimum energy barrier aspect of folding, that molecules like to avoid climbing high barriers as they move from one configuration to another. So I just want to give a couple of more examples to illustrate that concept because it's the basis for programming with folding pathways. Okay, so we'll go back to the two structures for the same sequence that I illustrated quite a while back. Two different structures. Imagine that the molecule is folded as on the left, and it's going to change its structure to reach the one on the right. Okay? So one way this could happen, I didn't want to draw out all the intermediate structures, but I think it's simple enough that you can get the idea. One way it could happen is that all those CG bonds break, and then the AT bonds get added one at a time. Okay? In fact, so that we would call a direct pathway, because the only base pairs that are involved along the pathway are either in the source or in the target. We'll see a different kind of example in a minute here. Okay. So what would the energy barrier of that pathway be? Well, initially the molecule has 4 base pairs, so its energy is -4 in our very simplistic model. As the CG base pairs are removed, the energy is going to go up until there's no base pair, the energy is 0, and then as the AT pairs get added, the energy goes down, back down to -4. So the gap between the initial or lowest energy and the highest energy is 4. So this poor molecule has to climb an energy barrier 4 to get to its final configuration if it follows this direct pathway. Okay? However, the molecule could actually fold or change its structure in a slightly different way, which I hope I'll be able to convey. And this is an indirect pathway. So the first thing that happens is that topmost CG base pair does get broken. Then a new CG base pair gets added, and I indicated that by a dotted orange line. There was that extra little G over at the end there that you might have been wondering all this time what it was doing over there. So it's finally becoming useful now. Then the 3 remaining CG pairs get broken. And at that point, we can add 3 of the AT base pairs without violating our condition that we like to work with branched structures. Okay, we don't want to create complicated structures. At that point, if we want to add the 4th AT base pair, we have to remove that little indirect one that we added, and then we can add the last one. If we do things this way, because we kind of added that little orange extra base pair and kept it in during most of this process, the energy barrier is actually just 3 instead of 4. So we can get the energy barrier down a little bit in this example. Any questions? This is about as complicated as it's going to get, but it's a good time to ask if that was confusing. Okay, so again, the intuition is you want to follow a pathway where you remove base pairs and add base pairs, and you want to keep as many base pairs in your structure as you can as you're following that process. Okay. So that's what a folding pathway is. A folding pathway is a sequence of structural changes, and it's good to keep in mind that as molecules change their structure, they don't want to climb over high barriers. Okay, so with that sort of principle in mind, how would you program DNA folding pathways? Here's an example of a simple way to do it due to Niles Pierce and Robert Dirks at Caltech. And they called their method, a very simple method, the hybridization chain reaction. So roughly speaking, you've got 2 molecules depicted on the left. They're like hairpin molecules. The little black lines, again, are base pairs. And these 2 little hairpins are very stable. They have lots of base pairs. And so they don't have much incentive to interact with each other. If they wanted to completely change their structure and bond with each other, which they could do because there's a whole strip on one of the molecules that's labeled B. B, you can think of as the whole sequence of bases along there. It actually matches very nicely with a complement on the other thing. But in order for them to pair in the way on the right, all the bases of one hairpin would have to completely break. So the energy barrier would shoot way up before new base pairs could form. Okay? So these, in some sense, are in a metastable state. They're very happy the way they are. And to reach a different configuration would require a high energy barrier. They're not very unlikely to do it. Okay. But if you add this initiator strand, the picture changes completely. So this strand has a little piece on the bottom, A star, that's complementary to a sticky end A on one of the hairpins. So it can form base pairs. And once those form, the next part of the initiator strand, B star, is complementary to B. And it isn't the case in this situation that you have to remove all the hairpins base pairs before you can add any new ones. You can kind of imagine unzipping the hairpin and that simultaneously starting to zip up the purple and the green strands. Okay, so as you remove base pairs from the hairpin, you're adding base pairs between the purple and the green strand. So actually your energy barrier is extremely low. It's like 1 or something. Yeah?
In terms of dynamics, it behaves in a way like a random walk.
Yes, it behaves exactly like a random walk once that process gets going. So the zipping is sort of going like that.
So there's no way to encourage it to go more in one way?
There are actually ways to encourage it to go further in one way. One way is to use concentrations. If you have an awful lot of initiator strands and very few of the hairpins, the initiators are more, they kind of act like fuel, and they actually do propel bias the reaction in a certain way. Now, that's not so much the internal dynamics of the random walk, but the overall situation. The internal dynamics of the random walk, you couldn't bias it really one way or the other. But there's other factors that make it likely. I mean, it's a very quick process, and it's extremely likely to happen, so quickly.
If the B part was much longer, then the process would slow down very considerably.
Not that much. An unbiased random walk is still pretty fast. Yeah, so that's not a key issue. Yes?
So how realistic are these movements compared to real molecules? Because I'm sure these are based on simulations.
No, these were really synthesized in the lab, and they really work. Yeah. I'll show you something really cool about them in a minute. Okay, so this slide just, again, fills in the details here that you really can get from here to here with a low barrier pathway if you've got the initiator strand, but you couldn't in the first picture when the initiator wasn't there. Okay, so that suggests that these hairpins can do things conditional on their environment. If something is in their environment, they behave one way, and if the thing isn't in their environment, they behave a different way, which is kind of one of the beautiful things about folding pathways. So Dirks and Pierce designed this in 2004, and very recently they published a paper in which they showed how you could use this idea to kill cancer cells. And again, they really synthesized the molecules. They really inserted the molecules into cells, human tissue cells, and they observed that this idea does work. So building on the principles I just introduced, instead of an initiator strand, what you've got is a strand in a cancer cell, a cancer marker. Okay? So in a cancer cell, this particular sequence will exist, whereas in wild-type normal cells, it won't exist. Then this is like the initiator strand. It triggers the hairpins to open, and when they open, part of it binds to this initiator, cancer cell strand, and part of it just forms a double-stranded helix. That helix then becomes a template for molecules called protein kinase R to bind, and these protein kinase R molecules can halt protein synthesis and ultimately kill the cell. So this is a really cool application, I think, of hybridization, chain reaction. And it's worked at least in cells in a test tube. So if the cell isn't a cancerous cell, the molecules will stay in their metastable state and nothing will happen. So this is really cool, say, compared to chemotherapy, which is just going to blast all of your cells. It doesn't really care whether they're cancer cells or not. It could work. Okay, so I'll just show you really quickly another example of a little robot you can build out of folding pathways. This little robot has 2 feet, and this molecule here, this fuel molecule, can actually bind to the dangling piece on the left. When it does, it forces that left leg off using kind of the same process. There's a little toe hole that can bind on, then there's a random walk process that happens and ultimately kicks that leg off. That leg can then bind to the next hairpin because there's a few sticky end bases down at the bottom. And then the fuel can come along and move the leg again, and you get this little walker. It's pretty cool. So you can do all kinds of cool things with DNA folding pathways. In terms of research, again, there are the same kinds of questions that I introduced for prediction. Computational tools to predict, say, minimum energy barrier pathways are very interesting. One problem, for example, might be to just focus on direct pathways and figure out what is the direct pathway from one structure to another for a given strand. My student Chris, who's here today, and some other people in our lab, Yano and actually Leanne, who's also here, worked on this problem, and we were able to show that it is NP hard, and Chris developed some really cool graph matching techniques that will solve the problem quite well in practice, even though it's hard. We're also intrigued by pathways that aren't direct, but can put in these sort of indirect or temporary edges as the molecule folds from one structure to another. And biologists hadn't really ever concerned themselves with, well, would a molecule really use indirect edges in order to get from one structure to another? So actually, Leanne has found beautiful examples that show that molecules can fruitfully use these kinds of indirect edges, and in fact can use them more than once. So, sort of getting a little technical here, but this raises the question, well, how long could the minimum energy barrier pathway be from one structure to another? If you can use all these indirections, well, maybe the path could be really, really long, exponential length, and maybe the problem with predicting these could be p-space hard. So we don't know the answer to that, but we're working on it. It sounds kind of negative, but actually it could be really positive, because if you can encode all that complexity into the folding pathway of a strand, well, maybe then you can do interesting computations simply by letting a molecule fold. So that's one direction that we're starting to explore in our research. So here's the last direction that I want to talk about. Energy efficient computation, which was one of Edelman's motivations that I introduced at the very start of the talk. So for many decades, certainly people at IBM in the 70s were very interested in the limits to energy efficient computation. a guy called Charles Bennett, suggested that, in fact, there was no fundamental energy cost to computation as long as you didn't erase data. So again, Eric Winfrey on the right here, and two of his collaborators, Lulu Qian and David Soloveitchik, came up with a theoretical, in this case, design of a stack machine that, in principle, would be able to execute and use arbitrarily little energy per computation step. How am I doing for time? If I have enough time, I'll... 22. Good. So I can tell you a little bit about how you would use folding pathways to build a stack machine. Really exciting for me. All right, so if you want to build a stack machine, you're going to need to support stack operations, pushing stuff onto a stack, removing stuff off of the stack. Very intuitively, you can imagine using a polymer somehow to do that. The polymer gets longer when you put stuff on the stack and gets shorter when you take stuff off the stack. I'm not going to tell you any more about it than that. Okay? You're also going to need to support transitions. If your machine is in a given state S and symbol sigma is at the top of the stack, you should be able to move to some new state S prime and generate some new symbol that gets pushed down the stack. Okay? So you need to somehow be able to support these kinds of transitions. So Lulu and her collaborators thought about how would you do that in the following context. Imagine that you've got strands in your test tube, some strand denoting state S and some strand denoting symbol sigma, sort of representing the current state of affairs. And now you want this transition to happen, which means that in that test tube, the strand and S and symbol sigma must be consumed somehow, and new strands must be generated, S prime and sigma prime. So how might you do that using folding pathways? Well, Eric and many of these other collaborators have been thinking for quite a while, actually, about implementing abstract chemical reactions in folding pathways. So if you abstract away a little bit from the specifics of a stack machine transition, more abstractly what you want is a situation where you've got two reactants, X and Y. They might be the state and symbol. They get consumed and you produce two products, S prime and sigma prime, or in this case, A and B. So how would you do that using DNA folding pathways? So my next slide, I'll show you how. It's a little bit hairy, but it's the last hairy slide. Okay, so what you've got on the top left are your two strands, X and Y, which are kind of the meaningful strands from our point of view. We want to consume them, and we want to produce A and B on the bottom right. Now, what we're going to do is we're going to have all these sort of auxiliary molecules that will help us achieve this floating around in the test tube. And they're pretty much inert. They're not doing anything until A and B are produced. Okay? And so those other molecules are reacting and producing A and B basically by folding pathways where X binds, then Y binds, pushing off some fuel molecules. And then two additional fuel molecules come and kick off A and B, producing A and B. Okay, so that's how the stack machine transition might work. Okay, so questions that we're interested in arising from that work is whether their simulation speed can be improved, whether fuel molecules, the consumption of fuel molecules could be reduced, and whether a machine could be designed so that multiple copies could run in parallel in the same test tube. So a lot of issues with their simulation that we think would be interesting to address. Okay, so to summarize from the last part of the talk then, you can program DNA folding pathways to facilitate movement and responsiveness to the environment. DNA folding pathways could be useful for transport or conditional therapeutic regulation. We saw examples of that. There's lots of new research questions, for example, how to achieve energy-efficient computations with folding pathways, and much scope to develop better methods for pathway prediction and design. Okay, so in closing, I hope I've sort of covered what I promised in my abstract. How do we program with molecules using sequence, structure, and folding pathways? And why might we program molecules? We can understand and change our world at staggeringly small scales. There might be applications to disease diagnosis and therapeutics, and it broadens our view of computation. Okay, so to conclude, I'll just say that personally it's been really fun to be involved in the field right from the start and watch it grow over a 15-year period and to imagine where it's going to go in the future. It's exciting to see the next generation of computer scientists with wet labs. There are quite a few of them now. And molecular programmers working in departments like cancer biology or systems biology, designing DNA scaffolds to do interesting things in those fields. I particularly want to thank many people I've worked with on this work. I'll just especially call out the people who are here today, Holger, Leanne, and Chris. It's been really fun to work with all of these people over the years. The ones on the top left are students that Holger and I have co-supervised or I've supervised in many cases. Other UBC students who got involved in various ways. Faculty that I've worked with, and Lulu and Eric who have inspired much of the work I do. Thanks for listening. Questions? I guess we're running a bit late, but you asked a lot in the middle.
Oh, I certainly have one.
Uh-huh.
If there's nobody else. All right, so I'm wondering, whenever people talk about that, I find it highly fascinating, and I get the feeling that there is a huge breakthrough in the way we can manipulate living matter just around the corner. And I guess something that you said today about the cancer therapy example is probably the first example that gives me personally the feeling that this might now be really, really very close. So I'm wondering whether you feel the same way, or whether you think it's another 10 years plus until our growing ability to manipulate living matter in computational ways really leads to first revolutionary medical applications.
I think it's a ways away still.
Okay.
Yeah, so in particular, I guess I wanted to say, I realized I'm running late, is I think the key words here are that these techniques offer the promise. And that's as far as I'm willing to go right now. I don't think we're there yet, although I'm optimistic.
And we're talking about the DNA to predict structure. I mean, what is the sequence of a DNA for a specific structure that you target?
Mm-hmm.
Do you also consider optimization? These are the minimum structure of DNA or some type of DNA for that kind of thing, or generally?
We haven't thought about that, but it's a very interesting question. I mean, certainly it's interesting. If a molecule performs a function, it's very interesting to ask, well, could you strip away some of its features and still get the same function? We're interested in that kind of thing, for sure.
You may do a better job in that kind.
Yeah, no, it's true. There's just tons of things you could work on here. I have no idea about that one. Okay, well, again, since I'm running late, thank you all for coming. If you've got more questions, I'm happy to hang around and talk some more.
Thank you for the wonderful talk. It's all good. You allow me to find a question between in this hybridization chain reaction, at least the way we talked about it, there was no consideration of structure and support between particles and I kind of wondered if you designed this carefully, should be able to actually use this today. Never going to happen.
Yeah, don't worry. I won't be tickling at you and being like, take one of your straps a little more.
I just wondered, when you answered the question about the device that for, that would seem like one of the ways in which you might be with non-biasing itself, but maybe you have some and that, of course, is a situation all of a sudden, we're really particularly assuming that not all stacking counts the same, like stacking in the middle might count, stacking in the end, you might only speed it up as you get.
Now, in the
Yeah, I know that's kind of why I was talking about here. Probably and of course you would have to have a multi-molecular, I guess sort of not activity. And then you would have to have an energy model that accounts for the differences where the stacks are. Which would also be true.
Very interesting. So, thank you very much.
That video was amazing. And it's not just very instructional, but also exceedingly beautiful. You know,
I like this video.
But if you do stuff on scaffolding, seem to speak to a concept of possible hardware software.
Oh yeah, well, I like the term Condon about, to combine it with the directions at the end of the class.
yeah, exactly.
And this is good.
Really
That is
exploring in terms of trying to get a rigid environment in which lots of things stay stable and other things are information is flowing, but it's done in rigid environment. I mean, I guess that's what the scaffolding suggests. I
just finished off the math part of Maybe I'm too limited view of the media for computation.
The more sophisticated we are,
send a packet, they start the timer, and then you receive the packet, but then you don't start.
So a space station has a scaffold exactly the purpose. That's good. I wonder if linguists are watching this as closely as they need to. For example, semantic structures, which need to be folded into something better. They're pushed this way not by syntactic cues and triggers, and if it works, you get a good result. Has that analogy been exploited by linguists? Okay.
probably, they're thinking similar things.
But not drawing the connection. Well, it seems like the same things are happening, of course, it's just different levels, different instruments of manipulation. I mentioned the path following graph matching solution to that. Is that published somewhere?
It's linked on my webpage.
So I can find that on your page?
Yeah, it's on the top.
Yeah. Optimum minimal energy paths. I'm trying to do in a linguistic problem, so I hope I can explain your. The one part, I guess one fellow asked, is this like a random walk? And that implies sort of equal probable bidirectional things. You might come just right to the end of the solution and then just walk back again because.
You might, yeah.
So
that's right, but there, yeah, I mean, I guess I can explain that better. But ultimately, you get to a very favorable thing that happens.
I didn't even finish my last question.
Eventually, it will actually move on to it.
Right, you have gazillions of them doing it all at the same time,
Yeah.
right? So I guess what happens is that some of them will walk back and forth, right? That's okay because others don't, right? Or don't do it quite as well.
You can simulate annealing by reducing the temperature. You allow all sorts of wild things at the beginning, but you're latching and latching and latching to closer to the minimum energy states, and then you're done. So it's one directional because of that gross manipulation environment. And here there is something, I guess, that happens at the end, which is that sort of a latch, which holds.
Exactly.
Okay.
Totally.
So
Yes,
that,
exactly.
Unfortunately, you can't decrease the temperature here. I mean, you could, but then you would slow everything down, even the productive steps.
Well, yeah, we don't work like our brains. We don't start at 98.9 and then work down to 92. But the answer is we can only do that for time. But thank you very much.
When I heard that 22, I forgot that we were on the half message.
But I'm sure you would have gotten cues from the audience if you would have gotten very nervous about that. So I wouldn't worry. I think everybody was
Yeah, I
captivated.
think basically structure I can build on is.
Oh, yeah.
Mine too. Isn't that like, what?
Hi.
Canada.
Hey, Kevin, thanks for coming. I got your email. I haven't responded.
That's right.
It's tough. I mean, I think right now I'm kind of decided like I want to take on more students, but I'd be happy to talk to you.
Other people if they
Well,
can
Holger
do
would be a good person. Yeah.
What is it that you're planning to work on?
He's an undergraduate, one of our best students. I'm very interested in algorithms.
In
Yeah.
algorithms.
Again, fortunately.
Yeah, I think if you want to, we should probably chat. I mean, in my group, there's always things to do. Currently, we are not working a whole lot on bioinformatics problems. However, that doesn't have to remain that way. Currently, our focus is on what we call automatic algorithm design.
Do you really need this to like solidify everything? This is like the conclusion of you don't have it.
Direction, in my opinion. And in that context, there is just one of interest.
Everything comes together.
One that I read
Puts everything
before.
together.
We'll probably have a second one.
Yeah, see, like everything.
So,
is like,
if you want to chat about that, by all means, contact me, or maybe I can forward you contact information or something like that. I'd be more than happy to chat with you.
is this supposed to drive all those?
That would be good. I mean, I'm not a Canadian citizen, so I can't give the answer.
Well, yeah, we'll have to look a little bit more into it. Currently, in my group, money is not the biggest problem in the world. So,
It's a good position to be in.
yes, it is a good position to be in. We are currently just looking for particularly motivated and particularly talented students. Yeah, if you did that skill and you are particularly
Who knows
motivated.
you well? Will is your work group. Who recommended you for that program at CS?
This is the last thing. This is the last thing.
No, there was somebody I asked.
It's like record hands right here. See, there's
I think
nothing
I
else.
asked Will. One of the most technical top students. Oh, okay.
Oh, yeah, okay.
And I took free 20 minutes.
Yeah, so, yeah. Okay.
I came for 20 minutes later.
Oh, okay, good. So they're probably already on this.
Normally what we do when people are interested in working with us is that we have a little meeting. You would send us perhaps your transcript before because we kind of like to look at that. It's not the only factor that counts, but it's one of them that we look at. And then we'll make a decision. So if you're interested in that, learn a little bit more about what we're doing, you're more than welcome to contact me. Set up that one.
Really?
Yeah, maybe it should.
Yeah.
On your web.
Why turn the ones that are still
Now
left?
from your web or from my
It's very easy. You type HOOS, which is my last name, into Google.
Take one of each.
HOOS. And then, or just off the, I'm a CS faculty member, so you just find me on the computer science faculty page. Very nice chat.
Yeah, imagine it.
Yeah, you have double
Thanks.
All right, good. Thanks for coming.
Yeah, thank you. Thank you for the call.
Now, of course, you can change it when you want to work with them after all.
Oh, yeah, well, yeah, I
You
mean,
should have first tips.
they're kind of the new decision. Have to do that. When you get these emails, of course. I
No, well, particularly if it's really good students, for sure.
Exactly, but I have to go back sooner or later. It's hard.
Good. I was trying to get a hold of you earlier today. It would be fantastic, speaking of moving back and stuff,
Yeah.
it would be fantastic if I could get a tiny bit more of your
Yes,
time
absolutely.
soonish, because after the message that you sent me, was it yesterday or this morning? I think you sent me one yesterday.
Yeah.
I got a rather official looking one this morning
Oh, good.
from the office of the VP research. I also yesterday talked to Alan, like I said I would.
Yeah.
He was highly supportive of the whole thing,
Oh, good.
so that was
That's
very nice.
Yeah, because that's.
And maybe a few really interesting insights about things that he thought could be done, and they kind of matched up nicely with or so meetings I had already chatted with you about, but then there were some other ideas, some very concrete things, which I really thought were super useful for me to learn. And today I spoke with Amy, so you know, I mentioned this might be happening in the context of teaching. There's two outcomes of this. The first is that she also encouraged me, which I didn't quite expect, but that was particularly nice. So she thought that might, from her perspective, she knows this.
Yeah, I don't know. I'm saying that the person can do it, I probably could find it.
She said she would really like you and me and Ermi to sit together and contemplate the possibility of just not teaching it for 1 year, just as a fallback option. She said she doesn't have a problem sort of checking it out. What's this community that's living at? It's not classroom experience, but I get confused with that because it's changed so much. But she says there is a committee that should really decide whether changing 445 into the fall, even this year, would cause destruction of other courses to the students. I thought that wasn't the case, but I see why she wants to be careful. She said she has nothing against doing that, but she also wanted to just put that option on the table of simply skipping it 1 year if we don't find it. So I said I'll bring this back to you. We might want, because Ermi is bought into this as well, we should probably just have a little discussion. And she says if we want to move it, if we really want to make a priority to move it into the fall, we should act quickly.
It was very recent, so I'm going to say you're still interested.
Because you won't know until probably May.
Let's stay this
I don't feel like sacrificing. I feel like I'm marginally not making good on a promise that I made on 101, but Gail wasn't concerned about that, and it seems that you weren't super concerned about it.
I'm guessing you'll have much less competition than in game time, so we actually want to win.
How you feel then?
And they have to make bigger prizes, I think.
I mean, not offering
Sort of.
students that
You think so? Yeah. I wasn't sure about that competition.
many other things you could do is
You don't have to be in the
send out
location to
Exactly, which might actually also be a pretty safe thing to do, right? Because I don't mind if it's not me.
I don't know, don't you have it?
I'm perfectly happy if somebody else is doing it.
Right.
And there is a potential state of the world. I mentioned this to Ian because I've done quite a bit more thinking about it. You know, if this all unfolds, how would I actually do it? I think there is a way of thinking where I would.
Jonathan says he's going to go home right now and learn it pretty much.
The timing of that might be such that I might even come to the department and say, can I shift teaching into, you know, whatever.
Yeah.
Yeah.
Well,
Did you all
I
find
mean,
my work on
then
that?
maybe we should send out an email.
On the other hand, this one's even less competition.
It might be absolutely. I mean, Mirella, I just spoke with recently on Skype. She seems very stressed, unfortunately. But that was just a particular day, I suppose, right? So I guess I will want to part-time.
Okay, all right, yeah, that doesn't sound like, that's okay, I mean.
Yeah, I know, but it just sounded like something, I mean, I asked her for whether she wanted to do game work, you know, of a certain kind that I think are in software. She has a very easy for her to do. And she said, like, whoa, you know, I can't even think about anything about that kind of thing. I think we should still try, definitely, just, we shouldn't put all of our hope on her, probably, because she seems like a little
Okay, that's
uncertain.
good to know.
But nevertheless, I mean, I think we should do it.
So, yeah, we are interviewing for jobs. Don't know if you'll be around, and then.
I put it, there is some basic down there.
It's not so
I don't know
clear.
But anyway, we can both think a little bit
That
more.
makes sense.
Yeah, that's why I thought we should probably have a discussion about that.
My guess is that, you know, she still has that postdoc.
I guess it's good.
Under some pressure.
Well, they're all people smart.
Moving things along, so.
Otherwise, I'm working on that letter. I kind of wonder if I should probably reply to that official message from Terry. Or do you think I should just wait until that letter is done?
What's the popular complaint?
14 hours starting off.
I wasn't quite sure, you know, what the right strategy is. Should I just fire something quick back saying, that's fantastic, and yes,
Something
seriously,
I think that's the right.
official letter or something like
Yeah, I
that?
think that's the right.
And then follow up with the official letter?
That sounds really good.
Good. I just wanted to check that you thought it wasn't better to just do the letter quickly and send it
No,
off.
I think that's the right approach, yeah.
Good. And then,
And did he give you any, like, he did, yeah, like, the committee needs last week. So it seems like they're kind of trying to now get something more to put on the table. Right?
Yeah, I don't know. Okay, so that's good to know. I didn't know that they meant that it would
I
meet
think
next
it's
week.
next week, but I would say following, I wouldn't rush what you're trying to do. Right now, that's not so important. What's important is you feel happy with it because it makes sense.
Okay, that sounds good. And of course, I mean, you're kind of in a conflict position because you're the one who nominated me and kind of knows me well.
Yeah.
But now you're also sitting on this committee, right? So I suppose for you to see this letter and comment beforehand might not be the most appropriate thing. Or how do you see that?
It's a really good question because there's somebody else who's interested, just so you know, who's not in science, who's also sent me something. I'm kind of trying to select simply, how to give back to that person.
Yeah.
And I think I'm going to take what I'd be comfortable with, and hopefully this will be okay with you, is like anybody who asks me for input,
Right.
that I'd be happy to give it. I might not give it as much as I would.
Make sure not to discourage the new guy.
Won't say
The new guy might be better than all of us. I don't know what he posted out there.
We don't have a group.
I would give you this kind of feedback I might give you if I wasn't on.
Okay, so but you wouldn't feel uncomfortable with me showing this to you, and then you can decide what you want to comment on.
Yeah.
Sounds
Yeah, I
good.
think it makes sense, and I think as community members, it's again doing that at a certain level.
I don't watch other videos.
What I don't want to do, I want to do it in a balanced way. Obviously, I'm not going to, one person for one person or for everybody.
In a group, people do not necessarily be there for 48 hours.
Yeah, and maybe I should double check.
I don't know if I'm going to do that, so.
Personally.
Okay.
Some people do. But you don't have to.
Just
Yeah.
You can't. I mean, actually, in the worst case, if I happen to send it to you and it's just crossed, and at that point you decide that you don't want to comment on, that's still fine, right? Because it doesn't contaminate
Just doesn't
anything.
make any sense not to give any feedback to people who don't apply.
If I go, I'm not going to aim for the prize.
That sounds good. Otherwise, I'll
Sure, go
that
on.
out.
Try to make that work,
It says 3 there. I'm actually thinking of asking Terry whether that's a straight up limit or more guideline.
You could, yeah.
It's not Windows Phone to get connected.
Yes.
Yeah, that's kind of what I'm thinking. But I guess I can check this with him.
Okay.
Good. Sure. Thanks a lot. And see you.
What you are doing is like take everything, just understand the project. I don't know how to place
Thanks. How did it all go? How did it all go? I think it went all right. I went a little long, but other than that, I think it went okay. It was sort of like, can you talk like I completely did from scratch more or less?
We're just trying it out.
Yeah. But I'm
I thought there's like some copy, but
I just kind of want to know. I just want to get to
maybe not going up there.
They have a cool logo. Don't you want to work for a company that has a cool logo? Google has lots of logos. Google. No, but it changes. It changes like on a lot of days. Dynamic logo.
Microsoft, Apple,
Yeah, but it changes. Google's the most artistic. What's 144 times 4?
that's
376.
176.
Can't be, no, I said 144. Sorry.
Times 4.
Yes, right. Because 376.
600 something.
Alright, so
576.
alright, 5. I subtracted? What happened there? Like the less significant digits, right? Awesome.
You should pick a general relativity next year. I bet there'll be a lot of vector calculus in that course.
Maybe I should take that in graduate school too. I don't want to take too much next year. course that I want to take eventually, but you know, I think I took most of my electives this year.
You have the prereqs.
Except for some math electives.
General knowledge.
Yes. Actually, I think I could have taken it this year because, like, I know at least, like, I think the prereqs are sort of optional. It's just like recommended.
Some of them are recommended. Like
I think almost
like
all of them are
206
recommended. I probably could have taken it this year.
I think 206 is required. And then
I think 206 was changed to recommended.
I remember seeing 206 on there, 227 and 317.
Although taking it this year would probably have been a bad idea.
There was a talk on distinguished programming DNA molecules. Sounds cool.
for a class?
I assume I'm just here waiting for the Orbis session.
Oh, yeah. Great.
I assume that's you guys?
That is us. Correct.
Floppy drive.
Yeah, we can use a laptop board. We also have our presentation on boards.
Okay.
What's best?
Yeah, I think it might be best if we go down through a laptop.
Okay, sure.
CSQ will be coming?
Not off the top of my head.
No? No?
Just a little feedback. We should say what should be this.
Said first one?
I didn't see it on the posters that are on the door.
I think I can.
Yeah, no, it's it's
Is
on
in the upcoming event.
Yeah, it's also on the little sliders that
Yeah.
does.
Yeah.
It says there. I had to find one of those to find out what it was.
Okay, and then I had it up on the.
Yeah, so it should be okay.
Yeah, it should be okay.
Can do it. It's a beast, isn't it? That's a unique thing. Yeah. Oh, it's a universal data thing, is it? That's to push.
I think, is it easier that I can just put USB in? You have a computer here?
Yes, but it has a login password. So usually, it should, like all of those things, you just run up.
Oh, is that?
That's
And then we have the,
what is this then? That's It's a convert, it's just a converter, that's all it is. Yes,
So there's no power?
doesn't look like it. We don't have power.
And
Yes, we do. This is crazy. And so I can just, it's a universal adapter, so we've got like,
I run over here for this. And then there's another handle that plays on channel if you want. I don't have that channel.
That's your place. So that must be, that's
it should be working because people are pretty.
Is that normal?
Decent.
No. You're going to go have a meeting with one of my presentation and
with encryption.
yeah, like
Because the laptops are.
Yeah, that internet.
so after you shut it down, you can bring it back up.
Or
There are two of them.
Curious, yeah.
And the password changes too. And the same thing as.
Well, obviously, there's probably weaknesses in ways. So they're overtrained passwords, but they're not very good at being able to.
Well, actually, we should try and get code founder. Yeah, so that's what I'm saying.
It's pretty interesting.
okay, this room is very good in terms of our projection, so it should not be a problem.
That's what it is. See that?
It's a secret. Yeah.
We can't trace.
Doesn't even take one.
No, it actually thinks exclusion does it.
I can show that.
It's all that mental.
Yeah, the old one was
It's on the disk. I'm just going to run the check.
Yeah.
It's good to see you.
Yeah.
Yeah. I will be here. Yeah, yes. See you soon.
Seriously, was that kind of
Like British.
Yeah.
Royalty.
Yeah.
British royalty.
Someone Caucasian.
And you do the photo, you drink your tea, and you have your pinky up.
I was just talking with this guy the other night, and he said, I've worked in London for 20 years, and one day, someone asked my first apparently he lost his wallet, and he was walking in the shop. And I felt that this guy's going to be
It's very
he's gonna find it out.
Currently, it's on both.
He was just very good at all.
Do you like
We
it?
got him in the chase.
Yes.
Okay, let's choose that. If they can choose.
Well, he was just trying to say that you visit into a part of the culture that they can, you can do it at any time of day. And I don't want to, I don't think so. So I only drink tea, and I always prefer to have a candy cookie, sometimes with it.
No, but 5:30, we say 5:30.
Yeah.
That is it.
We can even start later.
Yeah, yeah.
So when's the cup coming?
Yeah.
So it's like a
Sorry?
Is that
No, it's
It's one of the best ones you guys wanted more for this stuff. It's here for your
Really? Wow. That's cool. So you like the chocolate cake? Oh, that's awesome. That's great.
Yeah, I take it back. It is on there, but it's usually at the bottom. I'm just crazy. Yeah, sorry, my mistake.
Oh, don't worry. Good. Surviving. We have so many. Yeah.
Good market recovery.
Keeps going on.
Yeah, I was actually going to say, like, I extended that graduation because they were saying last year was not such a good year to be in the job market. But now I'm happy that you're saying it's your year. Yeah. Finally, yes. To make that finally work there.
All the MIs event, JB sent it out, right?
I think they got it.
They got it.
I was just walking by and KJ was here.
Yeah, we bumped each other.
Oh, I see. So, but you got my email, right?
When does this go from the computer science side?
From
Site sometimes takes a while for them to send it.
When does this go?
Mark, how are you doing? Good to see you. How was the Yeah, very productive. Hi. Yeah, we met before. Yeah, you were at the fair. Yeah. And I didn't think you were at the fair. No, nice to meet you. How was it? Was it good? Yeah. One after the other. Oh, yeah.
Sometimes it's so difficult because
I think you had another colleague You had another colleague
Right, so we had Melody. Oh, Melody, thank you.
They told me together.
this test.
Are you seeing? Well, I have to. I'm seeing it on your
So who's the president?
That's the other one's on. You find so this is just
Peter Chung was the first. I think Peter, no, Viola was the first, but now Viola's coming.
Okay, so I should have.
Smart guy?
I think he's
He's very small, right?
No, he's going off. I don't know. I guess you can tell him.
If you're looking for an
I'm
happy. I'm happy. I said that the other day. John Christopher came here.
Sit on.
I remember that time. You were an engineer. Why did you an founder? But they're dealing with the business.
With their heroes.
What about your? Yeah, I guess oh yes. I mean, they're treating it like from students.
You play with numbers, they play with the code. They keep me going.
Like it's a huge detractor to things, especially if.
Yeah, just trying. I think it's a little conservative. Something to keep in mind. Exactly.
Well, if you want to draw.
Are there copies or
Just show off. You can come.
because catering usually comes in trouble. Yeah. And it's usually hard for them to wait and
Yeah, they want to take a name out.
wait, you have your card? I don't think
But it's like this.
Thank you. Is it full? I think it's small. Because I know the way that they are doing it, UBC catering, they are coming and they did before the function, they don't really come so like after the event, they host. So if you are here, by invitation, let's say I offer this
That seems like a very good way to
Hold on, your card.
Like sometimes kids pressurize things, recognize the best.
So that would be Ian? Yeah. Yeah, okay. That's Jay.
Students go
No, it was actually, I knew it's Jay. Jay or it's letter. Yeah. Which I know actually does.
So naturally, we kind of
that way it benefits me, like even if
Nice meeting you, Paul.
where you want to
Nice to see you.
I think it makes you guys think harder and work harder.
Let's see if I can find copies.
Where
did Marco?
I think he went through for the, they're supposed to bring through as well.
So you can freeze, there's lots of food, lots of
So, what is the format? Do you want to get them something first and talk, or?
That was the plan.
That was the plan.
Whatever we're waiting for the visuals.
Yeah, maybe I would find out.
I would cut them first too.
I don't have anything on that part, that's all.
Yeah, something.
Yes.
And then everyone was with.
Awesome.
So from what you need to next, and this side of campus is on the south side.
Right.
So, like, at the very end. So I'll go and check.
Oh, yeah. Yeah, I think we got this is a lot. That's good. Yeah. Situations change.
I'm saying that's.
You're asking me to do that.
And yesterday you said if you leave, the office becomes